Thanks a lot. I've been fortunate I cannot be there with you. I think this is a wonderful school, and a lot of credit goes to Ausra and Maria Lena, who started this, I guess, 10 years ago, and for all the people who have done all this great work, including Daniel, to sort of keep this going. And it also strikes me now that at the moment, I'm glad that I think all the Nordic Baltic things are, well, I'm glad that we're all members of NATO, to put it that way. So that seems to be a good idea at the time. So I very much believe in the Nordic Baltic idea. Okay, so what I'm going to give like an overview talk about, in some sense, why we do modeling. That's sort of like my perspective on that. And I'd like to start with this image from of Norway's most famous mathematician, Abel. And he, you can see in the lower right corner here that he died 27 years old in this miserable farm in south of Norway of tuberculosis. And in Norway, we don't die of tuberculosis anymore, fortunately, and also in the rich part of the world, we typically don't. And he was very upset. He was very upset when he was lying on his deathbed. I mean, he had figured out all these difficult mathematical problems, and these silly doctors couldn't even figure out the cure for tuberculosis. So he thought it was completely unnecessary to die of tuberculosis. And of course, now if you go to a doctor in, say, in the rich part of the world and come back with a problem with the brain, whether it's neurological, or dementia or psychiatric, it's not so much we can do. And it's, let's see. And this was also commented on in this perspective paper, like 10 years old now. It's just to say this was a previous director of the National Institute of Mental Health in the US, probably funding a lot of research on mental illness. And he's essentially saying, why are we making so little progress on this? Why aren't we doing better with all these resources putting into this research? And if you ask neuroscientists, why are we doing so poorly? They maybe say, they point to the different scales. There are all these different scales in the brain, which are intimately connected. I mean, in this school, you have learned about the modeling, well, at least both inside cells and neurons and networks and whole systems. And they have all these scales, which are both in space and time, and they are intimately connected. And typically neuroscientists have worked on one scale and have not been sort of working so much trying to bridge scales, maybe because it's been considered to be too difficult. But then you can ask, well, why is it so hard to bridge scales? Or are there other endeavors, like research endeavors, where they have to deal with all these scales and have been doing better than neuroscience? And I think there is. My favorite is weather prediction. I mean, weather prediction is this very multi-scale problem where phenomena at like a 10-meter length scale adds up to affecting weather at like the hundreds of kilometer time scale. So there's all these special scales involved. And this guy, Wilhelm Bjerknes, he was this meteorologist and physicist and said that, well, 100 years ago, he said, like, weather prediction is a mathematical problem. What he meant is that if you know the weather today, meaning pressure, temperature, winds, and everything, then if you know the laws of physics that sort of determines how weather develops, so it's essentially just a big calculation. And of course, at that time, it was difficult to do, because it was sort of to pursue in practice because they didn't have computers. But with the advent of computers, this became a practical thing. And now all weather forecasts are computed from such models. This is the weather prediction for Krakow that I printed out yesterday. I'm not quite sure how accurate it is, but it's really been shown. It seems like it has a wet weather coming. I don't know, but it really seems that I mean, well, if you measure it, you can compare predictions with the weather that actually turn out, and you can see that your doing quite well, and not only doing quite well, you're doing better and better as these techniques develop. They say that I read this paper from 5 years ago, where they said that the inequality of weather prediction is steadily increasing. You predict 1 day better into the future per decade, meaning that the accuracy of predicting 6 days ahead today is the same as the accuracy of predicting 5 days into the future 10 years ago. This is quite amazing. And I think this is one of the most impressive things that natural science have been, achieving the last decades. And it's interesting also that nobody has heard about the famous meteorologists, right? It's just like the way they are able to collaborate and gather data and so as I said, it has been a quiet revolution with no famous heroes. So I think this is something we can sort of take heart from and maybe say, well, maybe it's possible to do this kind of bridging scales in neuroscience also. Another example is the smartphone. Smartphone is a different thing because it's man-made, but nevertheless, you have to bridge all these scales going up from the atom all the way up to the smartphone, just like you have all these scales going up from the atom to the brain. And how has sort of we, meaning mankind, humankind, been able to make smartphones? No single person in the world can make a smartphone. It's a combination of people. I mean, it's been a combination of scientists and engineers and also lately programmers that's been able to make a functioning smartphone. So, and how have they been able to collaborate, communicate so closely? Well, it's been through, they have found this really good language, which is mathematics, right? That's really the only language which is precise enough that it can sort of convey information between different scales so that it could together make something work. There's also now a new level actually of computer programs where you have many scales of programs. You have some people at the like who can compute, I guess, in hardware and all these zeros and ones and all this like machine code. While when I'm programming with my smartphone, I'm just putting my finger on an app icon, and suddenly I get what I want. Maybe like the weather in Krakow. So and I think there's like 4 levels of coding building on top of each other, so that we can sort of these scales in the program has been sort of been crossed. So I first want to say a little bit about, before I go into this, how to take this into neuroscience, that there's about the types of brain modeling. I remember when I used to do semiconductor physics back in the days before I got into neuroscience, which is of course much more interesting. But also when I went to these computational neuroscience conferences, I got confused because there's so many ways. There are many more ways to model brain systems, and principally different ways to model brain systems than there are, for example, for like semiconductors. There, everybody was sort of essentially doing the same thing. So, like one way to organize it is from like bottom-up type modeling and top-down type modeling. And I think with bottom-up, this has been the main topic of this course, at least in sort of like an amount of lectures. I haven't been able to. I've been attending quite a few lectures, while watching them on video, but not all. But I mean, typical examples are like how to modeling intracellular pathways, or neurons or networks of neurons, using tools like Neuron Nest and the virtual brain, which has been taught in this course. And then another type of modeling, which actually has been more prevalent, at least historically, has been what's called statistical or descriptive modeling, where the goal is to account for neural data, for example, spikes recorded from neurons with a sharp electrode, but account for them in a mathematically compact way. And I think the most prominent example, I'm not quite sure if this was covered in this course, is the Jubel and Wiesel, who measured these receptive fields of neurons in the primary visual cortex and found that they responded most actively to sort of like elongated stimuli and often moving in certain directions and so on. And this property was accounted for by a mathematical function, the typical Gabor functions, which was a practical way to represent this data, and also represent them in such a way that you could actually predict the responses for the same neuron to other types of stimuli. So one example is receptive fields. Now, I think the last decade there's been a lot of interest in neural manifolds, where they actually measure, they take data from spike data from many neurons at the same time, and do some kind of like analysis with a particular kind of analysis, so that they can plot sort of like the network behavior in certain way, and represent sort of the network behavior in this low dimensional space manifolds. But now there's also, like the last talk by Buddhist was more on the type of modeling that you don't see at all well, they only see in behaviorals, in biological systems, or in systems with the brain. This, I would say, like this cognitive behavior modeling, where one tries to account for, and I would say like make models for, like phenomenological models for how animals learn and so on. And for example, a prominent example, and one of the, I would say, if you ask me, even though this is far from my expertise, it's one of the coolest successes of neuroscience, and also computational neuroscience, is sort of to be able to understand this reinforcement learning. Then you also have types of brain modeling at this like intermediate stage, and I think I heard to listen to this talk by Friedemann Senke early in the course, and an example of an intermediate modeling style is sort of what he presented there, where he sort of found way of training deep networks to behave in certain ways, but instead of using firing rate neurons, which is typically used in normal deep learning, he used spiking neurons, which are closer to how real brains, well, if the real brains implement this idea, it has to be with spiking neurons, because that's what neurons in cortex do. So this is sort of both, I would say, like a combination of top-down, meaning that is learning and so on, but also bottom-up, since it takes into account, try to model it with spiking neurons. There's no like right way to, at least the way I look at it, there's no right or wrong way to model. But what I want to talk about here is sort of this, I would say, physics type mechanistic modeling. So, and the good thing is that I would say that the principles for the physics type brain modeling is quite well established. Here there are like 7 books, and well, they have become sort of like textbook material. So we sort of know how to model, in principle, how to model the neurons. What we don't know is, I would say, like all the parameters going in. We don't have ways to sort of to construct, say, network models networks of neurons, model for networks of neurons, because we don't really have the information about how they are connected in detail, and you also don't have information about all the parameters that goes into the network. They are in a real biological, in real brains, these are tuned in a certain way, and we just don't know these tuning rules yet. So even though we know in principle how to model, we don't really know how to do it in practice, or to make contact between, say, network models and specific networks in the brain. So this is, but again, we have all this, we know how to model this, I would say, like the building blocks or the constituents and dynamics of the constituents at the different levels. We haven't talked about like how to model how molecules are hooked up by essentially by like this chemical bonds described by the Schrödinger equation. There's been lectures on how to model the signaling pathways with its rate equations, and how to model neurons, and also modeling neural circuits, and well, there's like different levels of coarse grainness that you use to model networks. So how are we doing? Well, I think the greatest success in computational neuroscience, like the physics type modeling of neural system, is the establishment of the Hodgkin-Huxley equations, which I'm quite sure was covered in this course, where we're able to find a way to make a physics-based model based on like cellular membranes, currents going in and out of the cell, and that was able to quantitatively account for the generation and propagation of action potentials. And now, at the time of Hodgkin-Huxley, they didn't know what these ion channels really were. Now we do. It's like proteins it's like tunnels made by proteins going up and crossing the membrane. And now there's like a separate new, or a project that has been going on for some decades, is sort of to understand how these sort of statistical descriptions of like how these conductances of these ion channels, sodium and potassium, how they come about from the physical properties of the proteins and the ions. Because Hodgkin-Huxley just assumed, or they fitted this to the experimental data, while now we can actually compute them at least in principle. So I think it also explains this. This is an example of this interaction between statistical models and mechanistic models. For the Hodgkin-Huxley models, the conductances were modeled statistically and was used as input to a mechanistic model. And now we can sort of use this, what's called molecular dynamics at the level below, to do like physics type mechanistic modeling of why these conductances are as they are. So this is for the good news. For the bad news, well, no, actually, still more good news, just illustrating how we can now use this type of modeling to compute. I guess this is 5 neurons or that receives input, and what the color coded here is the membrane potential, and you can see sometimes they flash when they fire action potentials. So this just illustrates that we can make our own little model world and simulate neural activity using these laws that was essentially found by Hodgkin-Huxley and then extended to whole neurons in the decades following. But now for where we're struggling, and that is sort of going from the neuron to the neural circuits. And it's not because we don't really know, we don't have models for synaptic currents or things like that. It's just that there's this complexity of unknown, all these parameters which are unknown. And another complexity is that if you look at the Hodgkin-Huxley model, you could actually, now with a simple computer like you probably did in this course, you could actually simulate the model on a normal computer. But if you want to, I mean, a human brain has about 100 billion neurons. A mouse brain has maybe like a factor 1,000 smaller, 100 million. But it's so. And even at the moment, you don't really know how to make a model of the mouse brain with 100 billion or 100 million different neurons connecting up and so on. But even if we did, we wouldn't have the resources or computational possibility to simulate it. So we need so that's like two things. We don't really know how to make the model, and we're not able to simulate models which are large enough or at least not as large as we would like. Now it's typically maybe up to 100,000 neurons you can do, but then it starts getting inconvenient and you need supercomputers, and you can do it. It takes a lot of time from you start the model to get the result back, so it's difficult to do research on it. But so that's why there's been several large initiatives to try to build I would say, go towards networks. They have a little bit of different goals and approaches. In the Human Brain Project, where I've been a part since for the last 10 years, since it started, the main goal, or at least a main goal, is just to make a simulation infrastructure, meaning making it possible to run models with say 1 million neurons eventually, or maybe even approaching the whole like mouse cortex. I guess that's 17 million neurons or something. So that's been focused more on the infrastructure. It's more like an IT project. The Mindscope project at the Allen Institute of Brain Science has been focusing on a particular system, mapping out all sort of like connections and making models for all these mouse neurons, building mouse network models, and mapping out both anatomy and physiology of the mouse visual cortex. I think this is a really, really cool project because they've been able to, because of the funding, which was from this Paul Allen, which was one of the guys who got really, really rich from starting Microsoft with Bill Gates. So he was able to sort of make this industry style approach to say recording mice, recording from mice both like physiologically and anatomically, so that we get much more data. And not only that we get much more data, he has, well, from the start, he died a few years ago, unfortunately, he has been enforcing that all data are publicly available. And not only that they are in principle publicly available, they put a lot of work into making these so that people actually can use it. And I don't know, I've been a modeler for a long time. I've always been sort of relying on getting the data from somewhere else. And it's sort of typically it's been very difficult to get data from university labs. Even though they sort of maybe promised in their grant applications to give out the data, it's like they don't really have the resources, or maybe don't prioritize to make them available. So in practice, it's been a little bit ironic in the sense that the only data that has been in practice available, at least of this type, for the community, has been funded by a private person, Paul Allen, while the publicly funded research has not been, or data has not been sort of made available. So anyway, so this is, it has partly to do with practical things and partly to do with the culture that some people have been skeptical of sharing the data. A third project has been the Brain Project, which has focused a little bit more on, I would say, like technology for, new technology for recording from neurons. So back to this, why do we build these like large-scale models? And it's not because you think that if you somehow you just are able to put in about like 100,000 neurons together, or 100 million neurons together, or 17 million neurons together, mimicking a mouse brain, then suddenly we will understand how the mice, that sort of like the mouse genie will come out of the bottle. So this is, it's not, it has, the idea with the Human Brain Project has always been to make the infrastructure so that people can use this as a research tool. They can test out their own ideas and put in their own parameters and so on. So this was argued in this paper that we wrote, like many of us in the Human Brain Project wrote a few, I think it's 3 years ago, why we need to do brain simulations. And just to illustrate the point of them is, I'd like to sort of, I think this is a good analogy to going back to Newton. Because Newton, well, he did many things, but of course he had this fantastic idea about the law of gravity. So his idea about the movement or the law of gravity and the movement of planets was that, well, if there are two masses, like the sun and the earth, for example, they have forces working between them, and the force is inversely proportional to the square of the distance and the proportion of the masses and so on. And then he also had, well, okay, so if you have masses and forces acting on masses like the earth, how would that affect the earth's movement? So you need another law for that, and that's like F equals ma, and Newton's law, Newton's law of dynamics, like Newton's second law. And then, so Newton had this idea. So then how would you test it? It's not so easy. So in some sense, he said, well, if I'm just able to solve these equations, then I should be able to predict the planetary orbits. But it's not so easy, right? If you look at these, I would be able to just look at these and say, oh, this would mean elliptical orbits, like it turned out to be, right? In order to do that, you need to have he actually had to develop mathematics to test these ideas, to test what are if these are the this is my laws. What are the predictions? So in order to check whether his idea was right, he had to invent calculus, which we now, at least with those of us who study physics, and I guess, and certainly, well, most of engineering also, and most of natural scientists have to learn. So it's only with this that he was able to predict and come up with all these predictions, for example, about the planetary orbits, or that you have this, what's called ebb and flow, like on the water, and so on. All these things that made people believe in Newton's law. And it's the same thing with this, why we need large-scale simulations. It's not because they by themselves, well, they are sort of going to give you explain how the brain works. It's because it's a tool for testing out ideas. So it's in that sense, it's similar to calculus. Okay, so now a little bit more closer to home. If you want to make models, physics type models, you need to be able to compare with experiments. So, and what kind of experiments can you do? Say if you want to make a model or understand cortical activity. Well, you can put in a sharp electrode and measure either spikes, or if you have a hollow electrode and are really good, it's really good at what you're doing, you can maybe also measure the membrane potential, difference between the inside and outside of the cell. But there are other types of measurements. For example, now you have all this what's called multi-electrode contacts, where you measure the potentials at many different positions at the same time. And now we have like these electrodes like this, like the neuropixel electrodes, which is sometimes a little bit like the state of the art, that can measure from several hundred contacts at the same time. And of course, now if you have more of those neuropixel probes, for example, then you can get up to really large numbers of neurons that you can record from. Both the spikes, which is typically what is sort of been investigated, but also the low frequency part of the signal, what is called the local field potential. And then you have all these optical techniques where you essentially shine in light and you see what take a picture of what comes out, and then you can do all kinds of tricks and choose different kinds of light to shine in, and also different kinds of markers and molecules, so that you can, for example, measure the calcium concentration in individual neurons. And then at the systems level, you also can then you can also measure things like the potential at the cortical surface, and with ECOG, or you can measure the EEG, the potential at the electrical at the scalp, and you can also measure the magnetic field with MEG. Of course, the advantage with MEG and EEG is that they're non-invasive, so that you can you can also record from humans. Of course, we never put sharp recording, or you never essentially cut the skin on humans just for research purposes. It's always clinical research purposes. So now say if you want to have this, now we have all this, we have these principles for how to model neurons and networks. We have this thing, we look it up in textbooks. And now we have all these different measurements. What do you do? Well, I think, so this is just an example. If you want to make a model for a piece of barrel cortex, that's could have been any kind of cortex, but this is just barrel cortex is like a piece of cortex that essentially represents the activation of single whiskers on the snout of rodents. So say if you want to make a model for that, then you can, one way, there's different levels of accuracy. It's different levels of coarse grainness. And I think they all have their virtues. It's not like, I think at least, what one level is all you need, one level of coarse grainness. You have learned we're using neuron, how to model biophysically detailed neurons, and also in principle, how to make networks of those. And using tools like Neuron, I can also use something called Arbor. And then you can also model the same system with point neurons, like integrated fire type models, and using NEST. Or you can model it at the level of population firing rates, using, for example, the virtual brain. And one thing is that you would like these sort of, in some sense, you would like to level 2 to follow from level 1, that if you do some approximation, level 1 and so on, that you can end up with the same, when model for the same thing at level 2. Something that you can systematically reduce between these levels of coarse grainness, and the same between point neurons up to firing rate populations. But then, how do you compare this if you have these models, and how do you compare model predictions with what you measure? Historically or traditionally, you've been only compared with spikes, meaning that extracellular potential, action potentials from individual neurons. But there are much more data available: LFP, ECOG, EEG, MEG, and all these optical methods. And experimental data is always going to be the limiting factor. So what we would like is sort of to be able to predict all these other measurements from these models as well. And this is a different kind of modeling. It's sort of like what's called the measurement physics modeling. And this is something that we in our group in Norway has worked on for a long time. So we have developed this tool, LFPI, that can sort of compute. We are focusing essentially on these electrical things and magnetic things at the moment. But now, developing tools so that you can, from any models at any of these levels, you should be able to compute not only spikes, but also LFP, ECOG, EEG, and MEG, right? And then you can sort of have much more data to against which you can compare your models with. So what is this? Well, how do you do this physics type multimodal modeling, or like modeling the measurement physics? Well, if you look at a single neuron that you do, say, model it with a programmed neuron, then if you typically one models the membrane potentials and computes whether you have an action potential or not. But if you want to compute the spike, then what it really corresponds to is you can get it from the same kind of calculations, but then it's actually weighted. What the spikes actually is given physically as a weighted sum of the transmembrane currents in the soma region, and we know the weights. While the LFP, EEG, MEG contributions from the same neurons is still the weighted sum of transmembrane currents all over the neuron, but the weights will be different. It's not that the formula will be the same, but just because the measurement electrode is a different place, the signal will look different. And if you want to measure like what's called, if you have measurements of what's a voltage sensitive dye imaging, then it's an optical technique. Then you can model the signal as a weighted sum of the membrane potentials close to the cortical surface. So this is sort of what we have in, you need to work out these mathematical connections, do the measurement physics. And this is, the first version of LFPI came out into 2014. And the second version, it came out like it was a paper in 2018 or something. So of course, the whole idea here is that people shouldn't be able, this is sort of, people should be able to have cool ideas and not everyone who wants to compare with this kind of data should be able to go through the work that we did to make LFPI. There's no reason. We had to have some kind of division of labor. So that's why it's important to have tools, just as for the same reason we have tools for simulation of neurons, using neuron nest or the virtual brain. So this is an example. Let's see. So now let's see. So what I'm showing here is, this is a population of 10,000 biophysically detailed neuron models. And here, when I started again, you will actually see that it moves forward in time. And this black thing here actually shows where on these neurons the synaptic inputs are. So at this time point, the neuron gets input at around the soma. And then we can see actually the membrane potential is shown here. And we see that it's most active, meaning most depolarized, close at these lower dendrites. At the same time, we see that there's now actually starting to get some, for the local field potential, that's what you would measure at these different electrodes. Different electrodes here, and you can see at this time point where you have this dashed line, you start to get some, well, you have some negative signal close to the soma or at the depth of the soma, while you have some positive signal. At the EEG, actually, you see you have this is you have some weak positive signal, not so easy to see, but you'll see it when I start the video again, because now when I start it again, it will go into a phase where instead of having inputs at the bottom, you get inputs at the top. And then the whole thing reverses. And then you see that you get like activation. Now you start getting activation at the top. And if we stop again, now we can see that now there's a negativity at the top and the LFP and a positivity at the bottom. And the EEG has sort of flipped signs. Now it's negative. It's sort of essentially reflecting the sign of the LFP at the top, is the same kind of sign. And now you would think, well, what if we have this input at the same time all over the place? That would be, you would expect quite a signal. And you do when it comes to the membrane potential. So these neurons really gets activated. Look at that. And now the whole, it lights up, but there's very, very little LFP. And essentially very, very, very little EEG also. So even though this neuron, even though now we're not focusing on spikes here, but this population of neurons may fire action potentials as hell, but they would be invisible in terms of the EEG or the LFP, or almost invisible. So it just tells you that the EEG is not the direct measure of what goes on, like, for example, the firing activity. Firing of action potentials, and the same with the LFP. So these are the spikes and the LFP or EEG are complementary measures which both tell you something about the circuit, but not the same thing. So if we now just keep on running, I think this is, and you'll see that, well, now we're not even driving it, so now it's really, really boring. Anyway, so this is just an illustration that this is how we can sort of compute these different signals. Now, so far, I've been looking at the level, the most biophysically detailed level. And in fact, if you really want to be kosher about it or really want to do it properly, these extracellular potentials only exist in these multi-compartment neurons. If you have a single compartment neuron, like a point neuron, there's actually it doesn't generate extracellular signals at all. So if you only had point neurons in the brain, we would be like have no EEG at all. So in order to compute, but you still want to be able to compute these signals also from simulations at these levels of point neurons. And then we need to do some trick. And so this is something we developed. This was in collaboration with our friends in Jülich. And so there we used what's called just like sample, Markus Diesmann, one of our friends in Jülich. He and Potjans made this model of 80,000 spiking neurons mimicking 1 square millimeter piece of a visual cortex in cat. And then you can run that with using Nest, and you can get this 80,000 raster plots, essentially, telling about the spiking activity. And then the trick is that we now, in this scheme, we store all the spikes. A lot of spikes to store because it's 80,000 neurons, and then it's like each of them have a few spikes. But then after in the second simulation, we replace all of these dots with or these point neurons with biophysically detailed neurons, and then we rerun it using these spikes as input to synapses driving these neurons, and then we get an LFP. So here's an example where we sort of some animation of this, where we the animation is running a little bit fast, but let's see, we can stop it just before, because that, hey, help. Okay, so now at time 900, there's a spike or a volley of spikes coming into this network from thalamus. That was just how this simulation was set up. And then we see that we get this volley of activity across these different populations, starting at layer 4, excitatory population. So, well, I guess layer 4 inhibitory is equally fast. And then at the same time, we get this LFP. And then we can also then compute, well, if this was the only thing happening in the brain, what would the EEG look like? And then we use something called, in this case, called a four-sphere head model. And then we can compute signals, well, compute EEG at the top of the head. So, final level, at the level of firing rate populations, you also have to make a trick. And I'm not going to go into the detail here. We have a paper or preprint just out about how to use this hybrid trick to compute LFPs, EEGs, and MEG from population firing rate models. So I won't go into the detail. But a key thing here is that if you want to be able to compute signals from large, say EEG signals typically come from maybe in humans, like many, well, I don't know, 100 square centimeters of cortex, maybe not that much, but it sort of depends. And it really gets, if you use even with this hybrid trick, it really gets computationally expensive. But we have developed this what we call the kernel trick, where we, instead of having to deal, well, with the contributions from individual neurons, we have to deal with contributions from individual populations. So the computational cost scales with the number of populations squared, and not the number of neurons squared. Again, and this is just to illustrate that the brown thing, which is the approximation, actually is quite close to the black thing, which is sometimes the ground truth. So one, we have a collaboration with people at Allen to try to use this like approach to try to tune or to further develop their model that they came out with a model of the primary visual cortex in mouse based on all the data and the literature and so on. Model had about 250,000 neurons and really detailed, had 21 different neuron types, and they were sort of genetically identified, and there was like all kinds of different morphologies, and the models, we made models for them based on patch electrode recordings and so on. It's really state of the art in terms of bottom up modeling of cortical circuit models. So then they made this model and had it predict sort of this model could then, the first version, it could predict spike responses to different kinds of visual stimuli. And now in a collaboration with them, we are trying to develop this model further. I would say like tune the model to also be able to predict LFPs, the local field potentials. Because the good thing is that they in addition to making this model, they also at Allen made this fantastic data set of about 60 mice where they had these neuropixel probes, and they had this set of there was like standardized recording procedure, and all mice received sort of like the same kind of visual stimuli with movies and ratings and so on. And they measured the spikes and the local field potentials with 6 of these neuropixel probes. So it's really an enormous amount of data, and it's open for everyone to use. So it's really a fantastic gift to good community, I think. So now we're trying to, So then what we're trying to do then is, I know that's the wrong way, maybe. Anyway. Yeah, so, and this is just, again, a teaser. This is something we're working on, where this is the response for 5 animals to full field flashing spots, meaning that you have a no stimulus, and then at some time 0, you get this spot that covers the whole visual field. And here I could have plotted the local field potential, but instead I plot the current source density, which is something that is derived from the LFP, but the more local measure of like population activity. And if you have questions about this, Daniel is a super duper expert on this, so you can he can answer anything you are curious about. But the one thing we observe here is that, well, for some of these animals, the pattern of sinks and sources, that's sort of like something has to do with a CSD, is similar, but not similar, but not the same. And then you have animals which are just different. So this is also a question that we have to think about when we make a model. Should we make a model for an average animal or individual animals, or how do you sort of? There's a lot of questions to be discussed regarding what are we really making a model for. Here we actually have an example of where we look at it like some kind of animal average. This is the CSD pattern of an average animal. And here is the best tuned model, the best model that we're able to find. Well, when we try to sort of essentially make a model that mimic this experimental data as much as possible. And we see that it has some similarities, also has some differences. And an important point here is that this model has enormous amount of parameters, particularly connectivity parameters, just like you have in a deep network. But it's not very easy to train this model, even sort of to fit the parameters to make it look like the experimental thing. I mean, they say that, well, give me 5 parameters and you can fit an elephant or something, but that's not for mechanistic models, physics type models. That's more for descriptive models where you just fit the curve. Anyway, I won't go into that here. There's also a preprint out of bioRxiv that you can take a look on. So one thing we want to go with this is sort of to make what's called multi-purpose network models, not small, like not models that are like a low dimensional, meaning that there's like simple dynamical systems that maybe can explain one particular phenomenon, but actually have a model that can explain many things at the same time, different kinds of visual stimuli, different brain states, and so on. So we have these so far had these workshops, we call it like towards multi-purpose network models. And if you're interested in this, all the like lectures from these workshops are available on YouTube. So I just want to very briefly mention this because it was a nice lecture by Dirk Schubert where he talked about the...
Gaute, we need to be wrapping up.
Okay, I'll just wrapping up. I just mentioned that this genetic thing that Dirk mentioned, this is something now we are now starting to get. We are using information from genetic studies and GWAS studies to inform also this network. So we are able to also include that kind of data. And then I'll just finally measure that as an output from this eBrains, this human brain project. The goal is, or we're working on making this eBrains infrastructure so that people sitting at small universities or maybe in small houses with small computers, but with an internet connection, should be able to run large-scale network models and get access to data and everything using this eBrains infrastructure. So that is something I really encourage you to take a look into. It's going to be released sometimes next year when the Human Brain Project is over. So thanks for your attention.